You know, the food on your plate, it's right at the center of an incredible technological revolution. I mean, it's happening at a microscopic level, but it has the power to change our entire world. Today, we're going to unpack the secrets of this Omics Revolution and what it all means for the future of our food. Okay, let's get into it.
At its very core, agriculture has always been about answering this one big question. For thousands of years, we've been trying to get more from our land, right? We want bigger yields, better nutrition, and just stronger plants and animals. But the way we're answering that question now, well, it's changing, and it is changing fast.
So to really appreciate just how revolutionary this new stuff is, we kind of have to take a step back. For most of human history, the only answer we had was traditional breeding. This was the classic method, the OG toolkit for making our food better.
And this slide, it just lays it all out, the old way versus the new. For centuries, everything was based on the phenotype, which is really just a fancy word for what you could physically see. You'd pick the biggest bull or the plant that survived a drought and just hope their offspring got the good genes. It worked, sure, but man, it was slow, kind of like traveling by horse and buggy. And for things you couldn't see, like the quality of meat, it was a complete guessing game. The new way? It's all about the genotype. We're not just looking at the outside anymore, we're reading the actual genetic code. It's like we just ditched the horse and buggy for a rocket ship. It's faster, it's way more accurate, and it lets us pick the winners right from the start.
Yeah, this huge leap from phenotype to genotype, it wasn't a gradual shift. It was a full-on revolution. And it was all sparked by some absolutely game-changing technology.
I mean, just look at this timeline. The speed of this change is just breathtaking. Back in 2000, sequencing a single plant genome was this massive, monumental project. Just 5 years later, next-generation sequencing hits the scene, and bam, the cost of reading DNA just plummets. Fast forward to 2009, and we have the entire genome for cattle mapped out. A whole new universe of data opened up in less than a single decade. It's kind of like going from the Wright brothers' first flight to landing on the moon, all in the blink of an eye.
So what is this game-changing tech, this NGS? Well, imagine trying to read a gigantic library, but you have to do it one book at a time, page by page. That was the old way. NGS is like having millions of tiny, super fast readers all working at the same time, all in parallel, just devouring the entire library at once. That massive parallelization is what made sequencing an entire genome not just possible, but actually affordable and practical for scientists to do.
And this incredible technology didn't just give us more of the same old data. No, it opened up entirely new ways of looking at life itself. It gave us what we now call the modern Omics Toolkit. So here we are in this totally new era. We've cracked the code. We can literally read the entire blueprint of life. So the big question is, what's next? What can we actually build with all this information to understand how life really works? The best way to think about it is like a factory. Genomics is the master blueprint for the whole operation. That's the DNA. But a factory isn't building everything all at once, right? That's where transcriptomics comes in. It looks at the RNA, which are the active instructions being copied from the blueprint right this second. Then you have proteomics, which studies the proteins, the actual workers and machines being built from those instructions. And finally, metabolomics looks at the final products getting churned out by those machines. Every single omicsine layer gives us a real-time snapshot of what's really happening inside that factory.
Okay, so we've got this incredibly powerful new toolkit. That's cool, but how does all this microscopic data actually translate to the food on our dinner plates? Let's look at some real-world examples of how this is already changing the game.
One of the sharpest tools in this whole kit is something called a genome-wide association study, or just GWAS for short. And the idea behind it is actually brilliantly simple. You take thousands of plants or animals, scan their genomes for millions of tiny variations called SNPs. You can think of them like tiny one-letter typos in the genetic code. Then, you use some heavy-duty statistics to see which of those typos are consistently linked to a trait you care about, like disease resistance or higher crop yield.
And here is where the rubber really meets the road. In plants, GWAS has helped scientists find the specific genes that let them survive a drought. For rice, which is a staple food for billions of people, it's helping us breed for bigger grains and better quality. And in livestock, it's a total game changer. We can now select animals that are just naturally resistant to expensive diseases, or we can identify the best bulls for high-quality meat long before they're even fully grown. This all means healthier animals and better, more consistent food for us.
And look, this isn't just some far-off theory. As you can see right here, these methods are already being used successfully in some of the most important food crops on the entire planet. The key takeaway is this technology is already making a real,
tangible difference in our global food supply. But you know how it is. Every revolution brings its own set of new challenges, and the incredible success of the Omics Revolution has created a brand new one, biology's big data problem.
To put it simply, our ability to generate genetic information has massively outpaced our ability to store, manage, and actually analyze it. It's like we've built this giant fire hose of data, but we're still trying to drink from it with a teacup. We are literally drowning in information, and the next huge challenge is just learning how to swim.
And this right here is where the next chapter of the story is going to be written. It's not just about getting bigger hard drives. We need smarter software and, honestly, a whole new generation of scientists with the specialized skills to make sense of all this. We also have to solve what they call the phenotyping bottleneck. It's crazy, but it's now easier to read a plant's entire genetic code than it is to accurately measure how it grows in the real world. And the ultimate goal, the holy grail, it's integrating all those different omics layers, from the blueprint to the final product, to create a complete predictive model of life itself.
So this brings us to a final, pretty fascinating thought. For millennia, we've been working to improve our food through slow, careful observation. Now, we can read the source code of life with incredible speed and precision. And with new tools coming out all the time, we're not just reading the code anymore, we're learning how to rewrite it. So the question for our future is no longer just, can we do this, but what do we want to create? As we get better and better